We are now being recorded. So welcome to this 8th video teleconference for BIOL 105L YC. So if we take a look at the calendar, we are currently in sync with our schedule. In terms of exercises, we are doing exercise 9 this week, which is agarose gel electrophoresis. So you're expected to have viewed the videos already and that you're preparing your lab reports for submission already. So what are we expecting in the next 2 weeks? In terms of new videos posted for activities, there's none. Instead, it will be devoted for semifinals. And as usual, our semifinals will not be an exam, but a project. So in this video teleconference, I will already introduce to you what the project is. And our next lab exercises, exercise 10 and so on, if ever there's an 11, I forgot, are virtual laboratory exercises already. So meaning to say, I won't be demonstrating anymore the steps and the processes. Instead, you will be given links so that you can access the virtual laboratory in your own computers. Okay, so that's for the next exercises. So in this video teleconference, I'd like us to review again these three related exercises, 7, 8, and 9. They are all related. You cannot do one of them without doing the other. Their results need to be investigated in the subsequent experiment. So for example, when we isolated DNA, the isolated DNA is used in PCR, and whatever the product is in PCR is also the one that we are using for agarose gel electrophoresis. So as such, in this video teleconference, we will be reviewing the steps and what we're done in these exercises. And we're going to have a focus later on exercise 9. Okay, so first exercise 7, so we need to recall what we're done. Okay, so I divided this into two groups. Okay, so we have here the first part and then the second part. Who among the group can summarize to us what we're done in the parts boxed in red? Just a summary. Anyone? Again, anyone who can share or help us recall again what we're done in this first part of DNA extraction.
Excuse me, sir. Nawala imong kwan, sir, imong presentation, sir.
All right. Okay, on the first, what we're done first in extracting DNA from cheek cells. It's important to be able to recall at least.
Kwan, sir. Using sodium chloride solution. First it was gargled, sir, and then it was then put in a falcon tube, and in that tube, there are the cheek cell, cheek cells, sir, inside the falcon tube. And then it was then a portion of it was then microcentrifuged, sir, to pellet out the cells.
Okay, so in this first step, our objective is to form a pellet of cheek cells because that will be the source of our DNA, no? Human cheek cells. Okay, so now that we have the pellet of cheek cells, um, can anyone again from the group summarize what were done next after the pellet was obtained? Do not be afraid to speak up. There's no wrong answer, right answer. Um, we are just trying to recall, no, what we're done.
Hello, sir.
Yes,
The pellets were then mixed with um, tawag ito, um, I forgot
instagene
this
matrix.
instagene matrix, which acts as a chelating agent for the DNAs that was present in the pellet. It chelates the magnesium ions, which are um, cofactors for DNA, sir. The sample was then, uh, along with the instagene matrix, the sample was then put in a water bath at 60 degrees Celsius in order to incubate the cells for 10 minutes. After the first 5 minutes, it was vortexed and then continued on with the incubation. And then after 10 minutes of the water bath, it was then placed in a dry bath for 5 minutes to be incubated at 100 degrees Celsius.
Right. So those were correct, no? So again, the instagene matrix, no, the beads particularly, will sort of chelate or imprison the metallic ions that are present in our cheek cell samples, no, like the magnesium ion as you mentioned. Why? You also mentioned that these are cofactors of the enzymes that might cause the decay of DNA. Your example was the DNAs, no, the DNAs. So not the DNAs na plural DNA ha, DNAs, DNASE. So DNAs, these are enzymes. So the enzymes cannot act because there's the instagene matrix. And to break the cell so that the DNA will be free, we need to heat up the sample. So that's why we have the water bath and the dry bath, so that the cell breaks and the DNA is released. And at the same time, the cofactors of the enzymes are trapped by the instagene matrix. And so exercise 7 is done. We refrigerated our samples. Um, but essentially what we got there from the sample are these all of the 46 chromosomes from all of the cells, the cheek cells, were actually there in the sample. But there was only one particular gene that we will be working on. Okay? From which chromosome is that gene and what is the name of the gene?
Chromosome number 16, sir. Allogene.
All right. So this allogene is found in chromosome 16. It's a gene that is found in an intron. So meaning to say it is not translated into a useful protein. So meaning to say, for me, I can have it or I cannot have it, I'm still okay, right? So we're trying to isolate that from the sample, which is from my cells, okay? And try to find out if I am a homozygous positive for ALU, a homozygous negative for ALU, or a heterozygote for ALU. Okay, and then these are the lengths of the genes that we are going to be having if ever I will belong to any of those criteria that I mentioned. Okay, and in order to do that, we need to do PCR. Okay, so in PCR, we had these steps, no? Okay, so can anyone explain what happens during denaturation, annealing, and extension in PCR? You can just choose one of the steps here and then talk about it a little bit.
For denaturation, sir, essentially the DNA is heated at 94 degrees Celsius in order to separate the two strands of it, sir.
Okay, so this will unzip the two strands of the DNA, or the two anti-parallel strands. Okay, that's for denaturation. What about for annealing? Anyone again? What's the main?
Uh,
Okay,
for
go.
annealing, sir, the two oligosaccharide primers will sort of flank.
Oligonucleotide.
Oligonucleotide, sir. Primers will um, flank the two DNA strands which have been separated, and they stick to the other one sticks to the 5 prime end, and the other one sticks to the 3 prime end. Yeah.
All right, so that's correct. So this is for the primers, your DNA primers, no, the annealing portion. And then we have the extension. Okay, so anyone may answer again. What happens during extension?
Go answer. The Taq polymerase will, starting from the primers, the Taq polymerase will slowly add the nucleotides into the complementary strand. And yes, sir.
All right, now going to the 3 prime, now adding at the 3 prime end of the primers. Okay, so that's correct. So notice PCR is just DNA replication, di ba? You've learned about DNA replication, I think, in genetics already. So this one is like an in vitro DNA replication, but done in the laboratory, no? And the replication is repeated 40 times because we are um, after many copies of the target gene. So there's also an initial denaturation so that we're sure na nag-separate talaga lahat, no, ng mga DNA complementary strands. And then there is a final extension so that we are also sure that all of the strands have been completely extended. Right. So in drawing, this is the DNA, for example, and you can see that only a segment is our target. No, in our case, the allogene. So in order for that to be replicated, that segment, we need to first unzip the DNA. That's why we have denaturation at 94 degrees Celsius. And once the DNA is unzipped, our two primers, which are specially designed, will then stick to both ends, opposite ends of the two unzipped DNA strands. Okay, and then we have loose, or we have a hanging end here in the 3 prime. No, the 3 prime is ready to be extended. So this is in annealing. The primers anneal, and then during extension, with the help of Taq polymerase, which can withstand high temperatures, new nucleotides are added in the 3 prime end of the primers. And so you have now replicated allogenes, no, in this example. And then we do this for 40 times. and you can expect 2 to the power of 40 copies of our allogenes after that cycle, those cycles. Of course, this is going to be impossible without the use of a special machine called a thermal cycler and a chemical that is added into our DNA sample. And the chemical is a mixture of oligonucleotide primers, both forward and reverse. There needs to be nucleotides, the A, T, C, and G, no, should be present, many copies of it. And then magnesium ions to help as a cofactor for the Taq polymerase. And then you have the Taq polymerase itself, and you have a buffer. This is your master mix for your PCR. So the exact number, if we are more or less, if we are going to have 40 cycles, will be about 1 times 10 to the 12 ALU genes. No, so like the ones highlighted here in white. So notice it's really cut on the right ends, no, of the ALU genes, not like the others here which have unnecessary extensions. Right, so that's PCR. So PCR is practically gene amplification. So amplify, pinaparami. Okay. Now that we have amplified the gene of target, we can then analyze it using exercise 9 agarose gel electrophoresis. So in a way, exercise 9 is a qualitative test. Now we are trying to see if I am homozygous positive, homozygous negative, or heterozygous for the allogene. And so our samples were loaded into a gel, an agarose gel that we prepared. Okay, so now I need volunteers again to explain how the agarose gel was prepared.
Agarose gel, sir, was prepared with by 1% mass to volume ratio with agarose powder and the TIA buffer, sir. So about, I remember it was zero point 0.5 grams of agarose powder and 50 ml of the TAA buffer, sir.
All right, so that's exactly 1% mass per volume ratio. So zero point 0.5 is 1% of 50 ml of this. Okay, so why 50? Because that's the capacity of our mold, no? So it has to be 50. TAE stands for tris acetate EDTA, and it's our buffer as well in our electrophoresis later. And here it's acting as a solvent for our agarose gel. How did we then process or cook the gel? What are some of the things to remember when you are cooking the gel? Again, anyone may answer. What needs to be observed? How would you know that it's already cooked?
The
The
agarose
solution
powder
is
is uh,
Okay, thank you, both of
I
you. Sige,
I
anyone
kanala
else?
ani. Agarose powder is dissolved, sir, and the solution is transparent already.
All right, so that's correct. Thank you, both of you. So, alam nyo na hindi pa siya luto kasi parang cloudy pa, no? So, you really need to cook it. Lulutuin talaga. So, if you just observe the temperature there, that was around 300 degrees Celsius, and then there was a magnetic stirrer. So, you need to stir it always so that it will not burn the gel. And when the gel is cooked, as you mentioned, it should be completely transparent. All right, thank you. So now that the gel is cooked, we now have this next step, no? Boxed in red. So anyone from the group would like to explain to us what was done? what are the things that should be observed when doing the next steps here, the ones that are shown here in red.
Go on, sir. Before placing the solution to the mold, it was first cooled to about 60 degrees Celsius, and then the solution was then poured into the mold, and um, the gel comb was placed also um, into the mold, and then once it dried for about 20 minutes, it was then removed carefully. Sir, wala siya,
All
sir.
right. So thank you. So our gel mold, this is the mold, the one in the center. This one supports the mold. It's important. This is adjustable. That this is a level surface. So you have here a level to guide you to make sure that the surface is leveled, because we don't want a gel that's unevenly thick. And so one after pouring the cooled solution, 60 degrees Celsius, the comb is then placed immediately and then left there until the gel solidifies. And the sign is that the color of the gel changes again from transparent to cloudy, as you see here in the bottom. So once it's this color, we are sure that it has solidified. And then we can then remove the comb first. Okay, remove the comb and then loosen the screw here so that you can remove the gel mold, as we see here, the tray. Okay, so can anyone explain that what should be observed when putting the gel in the electrophoresis chamber?
The wells are facing the negative side, sir, of the chamber, sir.
All right, so the wells where we are going to load or put our DNA sample should be facing the negative electrode or near the negative electrode or the cathode. And the other end should be near the or towards the positive electrode or the anode. Okay, because we're expecting our DNA here to travel towards the positive electrode. Okay, so that's correct. So here, I think we sort of skipped a step. What needs to be added? No, because here we have the gel, it's still empty aside from the gel. What needs to be added then into the gel or into the chamber?
Trace acetate adta, sir.
All right. So if you notice here in the illustration on the right, this picture, there is a level actually here that says max. Okay, so this is the level, the volume of tris acetate adta that we have to fulfill so that we will have a correct setup. So this one will completely submerge the gel, including the wells of the gel. Only then will you be able to load the samples. So do you recall what was loaded in well number one?
The DNA ladder, sir.
All right, so that will be sort of our standard, supposedly. But we will see later on that it did not mark very well in the result. And then what about in wells 2, 3, and 4?
The controls, sir.
Okay, so in well 2, we had the plus plus control, positive homozygous. In well number 3, negative homozygous. And in well number 4, heterozygous. Did I say homozygous tong dalawa? Okay. And then, of course, in wells 3, 4, 5, 6, 7, 8. Sorry. 5, 6, 7, 8, 4 where the DNA samples, so the one that we got from the PCR. And when we loaded them, we mixed them with a dye that's blue in color. What's the name of the dye?
UV
UV.
dyes.
Okay, both of you are correct. So the UV dye, the function for that, of course, number one is you will be able to see what you are putting because it looks blue already. No, otherwise it will appear almost transparent and it will be very difficult to track. Number two is later on when we view this, no, as seen here in the bottom, it looks like it's effective, but this is not the final look. No, we need to view this under UV light in a dark room because the UV dye will actually sort of glow in the dark. You know, its position. The UV dye first and foremost is gonna stick to the DNA, and the DNA travels. And when you view it under UV light, it will glow in the dark. So we will know where the DNA is in the gel. So now we run the gel electrophoresis. Can anyone dictate to us the voltage, the current, and the time?
It was set for 30 minutes at 100 volts and 200 amperes, sir.
All right. So once that's done, once it's set, you then turn on the regulator, press run rather, and you will see that it's actually working because bubbles will appear at both electrodes, not the metals of the both of these electrodes. So we just wait for 30 minutes. If you have a time lapse, something, I think I had a short time lapse photo or video. You saw that the DNA sample actually moved. The dye at least moved from the positive side, sorry, from the negative side to the positive side. And so this is how it appears after gel electrophoresis. But as I told you, it doesn't guarantee that it looks blue that there's DNA there. We need to view this under UV light. No, so kaya hindi pa to ang final result. So we used the UV trans illuminator to do that. So this one is a picture not taken. I did not show the video anymore because UV is actually quite dangerous. Okay, this is real UV, and when you turn it on, you need to have protective gear. Unfortunately, we only had the lab gown, so it has to be done really fast. So this is a picture of the gel. Now this is our, look at this. Here it appears like there are only two bands. But if you actually view that under UV light, you will really see where the DNA samples are. So here we have the ladder, which I mentioned did not really appear as expected. So here we have the three controls, and here we have our DNA sample. So they appear to be all identical, more or less. The interpretation of this is, of course, based on what's shown here on the left, but I will not tell you, okay, what the interpretation is because that is still part of your lab report to be passed this Saturday. Okay, so I hope you got the connection between exercises 7, 8, and 9. Okay, they are really part of the same workflow. And your semifinals requirement is sort of a simulation of exercises 7, 8, and 9. Okay, so let's go to your semifinals requirement. But before that, any questions about exercises 7, 8, and 9 first? Questions, concerns, comments? So when you're doing research pala that involves DNA extraction, No, for example, in your thesis, hopefully, no, during your thesis time, wala na, pwede nang face-to-face. You can already work in the laboratory. You can apply what you learned, what you saw. You will be able to perform all of these, no, PCR plus gel electrophoresis. So that's a standard operating procedure for DNA analysis. Questions, concerns? What do you think will be the most challenging part among the three exercises if you were the one doing it? Can you imagine yourselves? Imagine lang, hanggang imagination lang muna tayo. If you're the one performing, which do you think is the most challenging part?
Sino ko ang kuan, sir? DNA extraction, sir.
Oh, the extraction? Okay.
If the DNA is like not, um, for example, if I can't extract the DNA properly, it will not um, be successful pod during PCR and in the kuan pod, uh, AGE, sir.
Yes, understandably. So, no, so pag wala ka ng in the first place, you didn't have any DNA to it na na-extract or you extracted the wrong DNA. Instead of cheek cells, it's from the food that you ate. Chicken DNA pala kasi kumain ka ng manok tapos hindi ka nag-toothbrush. That could be a difficulty. But to tell you the truth, no, the chemicals that we use are actually really designed for human cheek cell DNA isolation. So I understand that you said it could be the most challenging, but in reality, I'm sure if you did it yourself, it's actually just as fast as I showed you in the videos. So thank you for that. So those are those are the probable challenges. Anyone else? So one said it's the DNA extraction. What about the others? I'll tell you na lang based on experience. So the DNA extraction, based on my experience, no, is actually quite fast. Although if you are sensitive to laway, saliva, and all of those, you will be the one siguro na you will be uncomfortable all throughout because you are working with mouthwash. No, that's where the cheek cells are from. So it's quite fast. And it's fast to do. And it's the accuracy and the effectiveness na sure na merong DNA at the end is near 100%. Next, there's PCR. In the PCR, the challenge will be there are a lot of pipetting, no? And it needs to be done in a cold temperature. That's why you notice that our tubes are all sort of placed on ice because if the sample warms up, the enzymes can start acting up and destroy our sample. And the master mix also is quite challenging to prepare. Um, I did not show it anymore to you because usually in the lab technician prepares that already for the students. So that's challenging because you need to work fast with the pipetting. You need to remember there are several microcentrifuge tubes. No, potential mistake is you place the wrong sample in the wrong tube, etc. So that's the challenge there. And here in gel electrophoresis, the challenge is the loading of your sample into the wells. Okay. I'm not saying this is a challenge for all. Some of you might go through these quite easily, but there are some na korog og kamot, no? So when they insert the tip of the micropipette tip here, sometimes dili siya matunong sa well, and the samples will be dispensed all over the place. So it will be quite messy. So in the videos that I showed you, thankfully, I think I was able to dispense all of the samples correctly in all of the wells. But to tell you the truth, even some lab technicians are admittedly they don't want to load the sample and the gel kay kurog ilang kamot. So that's the challenge there. Another challenge is kani pag turn on mo ani, diba, there's a current. Sometimes it doesn't work right away. So you need to do problem solving, which among did I put the cover correctly? Did I set the voltage and the current correctly? So those are some of the challenges. All right. In the PCR pala, if your DNA sample did not amplify correctly, then there will be no bands that will be visible here in your AGE. All right, so with that, let's go to your semifinals project. So this is simulation. Your group isolated an unknown species of bacterium from one of the drinking fountains in Xavier University. You've decided to go an extra mile in identifying your specimen by using its DNA. So let's say you have extracted DNA already. Okay, so here on the left are the DNA or the genes, some of the genes that you extracted from your bacterium, from your specimen. We have two genes shown here. We have 16S rRNA gene. This is the coding strand. The gray nucleotides here are not part of the gene. No? They are just extenders. So do not include them. Do not be distracted by them. And then we have an antibiotic resistance gene coding strand also from your bacterium. And we have extenders here and the actual gene itself. So this is what you extracted from your bacterium, the one on the left. The one on the right is found in the bioinformatics database, meaning to say in the web for prokaryotes. And these are the candidate bacterial species for the specimen that you got from the drinking fountain. So we have Salmonella typhi. So this is the DNA sequence that is available from the database online. We have Escherichia coli, and then we have Pseudomonas aeruginosa. So your task is number one, to construct a schematic diagram of the steps that you should do from DNA extraction, so include DNA extraction, to whatever subsequent steps that you need to do, okay, in order to identify the genes, your specimen, the species of your specimen. Number two, indicate the specific primers, forward and reverse. So there are two primers dapat that you should use for the PCR. And should be specific, meaning to say you should give the sequence. And it should be at most 4 nucleotides in length. So hindi pwedeng 5. No, at most 4 lang as indicated here in the instruction. So you need to provide both forward and reverse primers. So where will you base your answer there? Somewhere here in this slide. So I will not tell you, of course, where. That's problem solving. And then number 3, finally, state the identity of your microbe. So what is the identity of the microbe that you isolated from the fountain, drinking fountain at Xavier? So again, this is a simulation. It's not an actual laboratory work, but there are certain information given here, such as DNA sequences, and you need to solve the problem on the identity of your microbe. So that is your semifinals project. It's due on April 3, 2021, Saturday. Black Saturday. Okay, so I will be posting this instruction on our course wall siguro next week, no? So that you can start working on it. Or you may also want to take pictures now. If you did so, that will also be possible. Okay, so any questions or concerns? Questions, concerns?
Ay sir, about atong kuan ba DNA ladder ngano di ito siya wala na klaro.
Okay, good question. This DNA ladder, we have been using this for atong the other batch, no, na nag-cell mall also. The result was also like this. Probably it did not pass PCR kasi, 'di ba? This primer, not primer, this ladder was simply added during the gel electrophoresis exercise. You notice? Sa PCR, ang naka-pass lang ng PCR are the 3 controls on our samples. So maybe the density or the amount of DNA in our ladder is not that concentrated, but it's also evident here on the result. That's my best answer for that.
Thank you sir.
Okay, any other questions? Maybe about the project or about the exercises? With regards to your grades, I haven't finished checking yet, no, all the lab reports. So your grades are not yet in for the midterm. But if you're also uh, enrolled in the lecture component of this class, the grades have already been passed. No, so I think I've already talked to you about it, that it's actually your raw scores. So it might seem that it might not be what you expected. It might be a lot lower than you expected, or maybe the opposite. Well, good for you if that's the case. But it's the raw scores, no? And in the final grades, of course, there are other considerations that I will be including for your final grades. So rest assured, not rest assured, my message is this: do not be discouraged by your midterm grade, because of course that is not the final grade. Okay, it will just tell you whether you're doing well or you need to improve still. Okay, no questions? You can always message me through eLearn if you have more questions. So maybe wala na, no? So maybe we can end now. Thank you very much for joining in this video teleconference. Without you joining, there will be no teleconference happening and no recording also. So thank you. You can view the recording of this later on. So with that, guys, have a productive day ahead, and you are now all dismissed.
Thank you
Thank you,
sir.
sir.
Thank you, sir. Thank you, sir.